Hi everyone, how you doing? We are going to be learning about meiosis today, and as you can see here, meiosis is the division of your sex cells. So that means either your eggs or your sperms, and they're going to be called gametes. And what we see here is that we're going to go through like we did with mitosis, but we're going to do it two times because we're going to do two different divisions. So up here, this is going to be interphase, and this is where we're doing the S phase. And then from here to here, this is meiosis 1. So I have prophase 1, metaphase 1, anaphase 1, and telophase 1. So there's a lot going on in this part of it. And then we have meiosis 2 over here. And now from meiosis 1, we did cytokinesis and we have two separate cells. But this then has to go through another division. So we have meiosis 2. So again, we're going through prophase 2, metaphase 2, anaphase 2, and telophase 2, and then we're going to go through cytokinesis. And what we end up with is what is called a haploid cell. So we started with a diploid cell, which means it has a total amount of chromosomes. And what we ended up with are haploid cells, and these are also called gametes. And they're haploids because it's going to be egg and sperm. So I need half of my chromosomes from my mom and half of the chromosomes from my dad. So when it comes together, I have the full amount of chromosomes, like I did when I first started up here. Okay, so we're going to start with vocabulary first. Let's see. So we're going to go. So now we're going over our vocabulary. The first thing you're filling in is we have meiosis, and this is the formation of gametes. And gametes are going to be your sex cells, like your egg and your sperm. So what happens is we're going to reduce the number of chromosomes in a cell and this is going to create genetic diversity. So we have a cell here, if you look in the front here, a diploid cell, and we'll get to what that means. But notice how it has 4 chromosomes, 2 blue and 2 red. So with meiosis, like I said, we go through 2 different divisions. So if we look at this second little diagram, it's going through the S phase, right? Because I now have 2 copies of each. And my first division, which is called meiosis 1, is going to separate those 2 into 2 different cells. And then in meiosis 2, we're going to go through a whole nother division, creating 4 haploid cells, and we'll talk about that in a second. But in this, it's going to create genetic diversity. So this means that you're not going to look like your siblings unless you're an identical twin. All right. So the next one we have is called haploid, a haploid cell. Oops. Okay, so a haploid cell is either an egg or a sperm, and egg and sperms are called gametes. But if you think of haploid, it kind of sounds like half, because haploid cells have half the amount of chromosomes. So it's going to be your egg or your sperm. So according to the notes, we have 1 copy of chromosomes. Gametes are haploid cells. So that means your egg and your sperm are haploid cells. And humans have 22 autosomes, which are like for your body and everything, chromosomes, and you have 1 pair of sex chromosomes, which are your gametes. And you can see here in this image, we have 4 cells because we started out with 1 diploid cell. Well, we'll get to that. Has the full amount. and we end up with 4 haploid cells, which is an egg or a sperm. And then when those come together, we're going to again have that full amount of chromosomes. So next we have the diploid cell, and again, this is 2N because it has twice as many chromosomes as it did in the haploid cell. So these are basically body cells, which are also called somatic cells. And you have 2 copies of chromosomes, like you see here in this diagram, of diploid cells. And this happens when you get your egg and your sperm together, which had half the amount of chromosomes, and they come together and they make a diploid cell, which would give us the complete, in humans' case, the 46. So I'd get 23 from mom and 23 from dad. They come together and I have 46 chromosomes, and that would be a diploid cell. Okay, next we have what's called homologous chromosomes, and I know that you should be familiar with this. I used this term before several times, and these are a pair of chromosomes that are similar but not identical. You inherit one from your mom and one from your dad. So these carry the same genes, although the genes may code for different traits. So for instance, on the chromosomes, let's say at one level, let's just pick something here, at one part, it's going to code for hair color. So it's going to code for hair color at the exact same spot on the other chromosome. It just one may code for blonde hair and one may code for brown hair. That's all that means. And then we have what's called synapses. Synapses is the pairing together of homologous chromosomes in meiosis. So what this means is I have this chromosome from mom and this chromosome from dad, and they're going to come together like this. And that's called synapses. And then what happens after that is our next vocabulary word, which is crossover. So while they snap together, what's going to happen is some parts of the chromosome from dad are going to swap with the chromosomes from mom. So this is the one that's creating genetic diversity, and it's very, very important. So this is what, again, causes genetic diversity. So this is why we don't look identical to our siblings unless you're an identical twin. So if you look over at this image, you can see how the synapses, they're coming together, but we can see in here where they exchange that genetic information between the chromosomes. Alrighty. Next we have gamete, and a gamete is another name for that sex cell, the egg and the sperm. So gametes have the DNA that will be passed off to their offspring. So we have over here an image of the egg and the sperm, and each of these only contain half the amount of chromosomes as a full-on cell, because when they come together, they're going to make a diploid cell. So at the moment, each of them are haploid cells. They have half the amount or one copy of the chromosomes, which is represented by N. And in humans, that number for N would be 23. So I'm going to get 23 pairs from mom, 23 pairs from dad, and then that will make a fertilized egg, which will give us then the 46 chromosomes. All right, the next one we have is called somatic cells. Somatic cells are not passed down to offspring. So in humans, each somatic cell has 2 copies of each chromosome. So that makes it 2N. So remember, N was 23, and 2N would be 46, the full number of chromosomes. So these are going to be every other cell in your body except your gametes, your egg and your sperm. So all your body cells are going to be this. Um, so these cells, these chromosomes will not be linked to what your sex is, for instance. Only um, uh, your gamete cells will be in charge of that. All the rest of them are just giving you your traits and everything. And that is your vocabulary words. And what you're going to do now is you are going to put together this diagram that you picked up. So there's this part, and then you have another one that goes to it. So you are going to um, glue these together so they match up down here. All right. I don't know how it matches up, but they do. They match up perfectly. Take that. Okay. It's always hard to tell which way is up. Okay, so you're going to do this. And what you're going to do is you're going to be filling this in, and I am going to now show you what this is going to look like. So let me make this a little larger. So this is your diagram of meiosis, and you're going to be filling this stuff in. So this first cell at the very top is a diploid cell. So it's 2N because it has all the chromosomes it needs, right? So, and you can see interphase G1. This is before DNA replication. And we have 2 pairs of homologous chromosomes, one from mom and one from dad. So that's where we got the 40, well, in humans it would be 46 chromosomes because 23 came from mom, 23 came from dad, and then they come together. So the next one I have here is interphase again, but this is the S phase. So this is where we have DNA replication. So if you look at this image, you can see now from the first one that we now have an extra, another copy to make that chromosomes, right? So they were sister chromatids. So now we're going to go to, let me just widen this. Now we're going to go to prophase 1. So before that, if you look on the side here, it says meiosis 1. Okay, so this is going to be the first part of the division. Next, I have meiosis 2, which is the second division. So in meiosis, I have 2 divisions. I have meiosis 1 and I have meiosis 2. So we're going to be talking about the first one. First, let me move this over here. So we have prophase, and this is really important because this gives us crossover, which gives us genetic diversity, and that's really, really important. So this is where synapses came into play and crossover. So they came together and then they exchanged DNA between them. So this is creating genetic diversity. It's extremely important. So crossover, prophase 1, genetic diversity. Key things to remember. In metaphase 1, and we call it 1 because it's part of meiosis 1. It's the first division. So we have independent assortment. So this is where our chromosomes are going to meet in the middle. But here we don't know, since we have this crossover, how exactly are they going to match up in the middle, right? So one chromosome could flip over and match with another one or whatever. We're not really sure. So if you look at the image here, it's not exactly like mitosis because we are just dividing the chromosomes, right? The full-on chromosomes. That's all we're doing in meiosis 1. So you can see from this image that it's lined up. I have 2, in this instance, I have 2 chromosomes here and 2 chromosomes here. And again, this will create genetic diversity or variation because one of these could have lined up with one of these other chromosomes. So it could line up in any way, which is again causing diversity. Then we have anaphase 1. Again, all we're doing is we are separating the chromosomes in meiosis 1. So here you can see in this image that I'm pulling the full chromosome apart from the other chromosome to opposite sides. You know, like anaphase, I pull apart, but I'm pulling the entire chromosome apart here. So you can see that. And then we go to telophase 1, and this is again where we're reforming our nuclear membrane, and we have our cleavage furrow, and this is resulting in 2 non-identical daughter cells. So these are genetically different from each other. All right, that's very important. So meiosis 1, we are separating the chromosomes, and then we're going to go to meiosis 2, and what we're going to be doing is then dividing the sister chromatids. So after telophase 1, we went through cytokinesis, and now I have these 2 cells. I don't know. And again, we're going to do PMAT again, or prophase, metaphase, anaphase, telophase, and then cytokinesis. So we're doing this all over again. But this time we are separating the chromatids, not the full chromosomes, because we already did that. So in this one, you can see how I have 2 cells now, right? This is the same process. Okay, so I go through prophase 2, that's indicating this is my second part of meiosis, meiosis 2. So in prophase 2, and this is going to be closer resembling mitosis. Okay, so prophase 2, my nuclear membranes are starting to dissolve. My spindle fibers are going to form in both of those cells, right? And then we go to metaphase 2. In metaphase 2, in both of the cells, we are lining up in the middle with the spindle fibers helping facilitate this. All right, so chromosomes line up in the middle, and there's still, again, more random genetic variation here because it depends on how we're lining up. That's going to make a difference. Some chromosomes could line up in one order and others in another order, so there's variation there as well. And then we get to anaphase, and here we're going to pull apart. So we're going to pull these chromosomes apart into sister chromatids. Okay, so now I have that, right? So then I go to telophase, and in telophase, the nuclear membranes are going to reform, my spindle fibers are going to disappear, and you will see the cleavage furrow, right? And these will now be creating haploid cells, which if you recall from your definitions that we did or vocabulary, that it has half the amount of chromosomes. So if we look at the bottom here, we can see that once we go through cytokinesis, we are going to have 4 genetically diverse haploid cells, which are gametes, which are sex cells. So in the instance in this diagram, they are sperm, right? So you're going to get 4 full-on sperm. For women with eggs, what happens is we're going to get 1 egg, big egg, and then we're going to get 3 what are called polar bodies. And these will just dissolve and go away. because they won't be usable because everything will be in that egg, that one big egg. So when we're doing sperm, I get 4 sperms. When I'm doing eggs, I get 1 major eggs and 3 polar bodies. So that's kind of important to remember. And that's it for right now. You can always stop along the way to write stuff down and everything. Thank you.